Hello students, good morning to all of you. Today we will study about the last plant of the gymnosperm, that is the Ephedra. The systematic position of the Ephedra, it belongs from the division Gnatophyta, class Ephedropsida, order Ephedrals, family Ephedraceae, genus Ephedra. The genus Ephedra includes about 42 species. They are widely distributed equally in the temperate and subtropical regions of eastern and western hemispheres of the world. In eastern hemisphere, they mainly occur in Mediterranean areas, France, Portugal, Spain, Germany, Italy, Israel, Iran, Afghanistan, Pakistan, China and India. In western hemisphere, they occur abundantly in the Andes region of South America, New Mexico, California, and North America. In India, about 8 species have been recorded. The distribution and diagnostic features of some common Indian species, that is, one is the Ephedra foliata. It represents the climbing species commonly occurs in deserts of Punjab, Sindh, and Rajasthan. Ephedra intermedia, it is service species commonly occur at an altitude from 2500 to 4200 meters in Pangi, Kashmir, Kullu, Jaunsar. Lower two-third bracts of female Strobilus are connate. Seeds are 5 to 6 millimeter long, tubulous, twisted. Ephedra giardiana, it is service species, commonly occurs at an altitude of about 3000 meters from Kashmir to Sikkim in dry alpine and temperate Himalayas. Strobilus 5 to 8 millimeter long. Seeds 4 to 6 seeds 4 to 6 millimeter long. Tubulous straight, not more than 1 millimeter long. The plant body of the Ephedra. The first we describe or discuss about the external features. Most of the species are small, xerophytic woody shrubs, rarely exceeding 2 meters in height. A few are prostrate. Many species spread by rhizomes from underground buds. Some species attain tree-like habit under favorable conditions. The plants are differentiated into root, stem, and leaves. The root is a long taproot characteristics of xerophytes. Adventitious roots may arise in larger forms. Next, that is the stem. The stem in Ephedra is woody, jointed, and profusely branched. The branches arise in the axils of scaly leaves. The young stem and branches are slender, fistular, green, and photosynthetic phylloclades. The old stem is woody. In some species, the rhizome may also be present. The jointed stem is differentiated into nodes and internodes. The internodes possesses basal meristematic zones. Activity of this meristem results in the elongation of the internodes. The meristematic zone dries up at the end of growing season, which causes brittleness and shedding of branches. Next crop of branches comes out in the next growing season. In the next growing season, profuse branching, reduced foliage leaves, and jointed nature of stem gives the plant as appearance of equistem. The plants are therefore commonly called jointed fir. Next, that is the leaves. The leaves are minute, scale-like, and borne in pairs at each node. They arise in opposite decussate manner and remain united at their base to form a basal sheath. In some species, they are arranged in whorls of three or rarely four at each node. Each scale leaf, each scale leaf is traversed by two unbranched parallel veins. The scale leaves are deciduous. Foliage leaves are absent, and the scale leaves are non-photosynthetic in nature. So the function of photosynthesis is carried by young green stem. Next, go to the internal structure or internal anatomy of the root, that is TS of the root. You see the figure. First we started from the outside, that is outermost layer, that is the epidermis. The outermost single layer epidermis consists of thin-walled parenchymatous cells. A few cells elongate to form root hairs. Inner to the epidermis, that is the cortex. The broad zone of cortex is differentiated into outer few layered collenchymatous outer cortex and inner multilayered parenchymatous inner cortex. The innermost layer of cortex forms endodermis, and endodermis is followed by single layered pericycle, which is consisting of thin walled cells. Inner to the pericycle, that is the vascular cylinder. Vascular cylinder consists of radial arrangement of xylem and phloem. The xylem is exarch and diarch. Sometimes it may be triarch. Next, go to the internal structure of stem. A transverse section of the stem or the primary structure of the stem without secondary growth. The outline of the section is irregular due to presence of ridges and furrows. In this structure also we are started from the outside to the inner side. In outside the epidermis layer is present. The epidermis is single layered, consists of thick walled cells. It is covered with a thick layer of cuticle. The stomata are haplocoelic, sunken, and embedded in circular pits. They are distributed in the region of Purroge, in inner to the epidermis, that is the hypodermis. In hypodermis, the patches of thick walled scamatous cells present below the ridges represent the hypodermis. Hypodermis provide mechanical strength to the plant. Inner to the hypodermis, that is the cortex. Cortex is differentiated into outer 2 to 3 layered outer cortex consisting of palisade like cells and inner 2 to 3 layered inner cortex consisting of spongy parenchyma. These cells are filled with chloroplasts and perform photosynthesis. These cells are separated by large intercellular spaces. The innermost layer of cortex forms ill-defined endermis, which is followed by single-layered pericycle. The pericycle is not well marked. Inner to the pericycle, that is the vascular cylinder. The stele is ectoploic cypnostele to U stele. Vascular bundles are arranged in one ring. Each internode possesses 8 vascular bundles. Two pairs of small vascular bundles representing leaf traces and 4 large vascular bundles. The vascular bundles are conjoined, collateral, open, and arch, which are separated by broad parenchymatous medullary ridge. The cambium present between the xylem and phloem is persistent. The xylem consists of tracheids and xylem parenchyma. The phloem consists of sieve tubes, phloem parenchyma, and albuminous cells. In center, that is the pith. The center is occupied by large parenchymatous pith. In nodal region, the pith is composed of lignified cells which forms the nodal diaphragm. Next, that is the secondary growth. The secondary growth is normal in the Ephedra stem. It begins with the formation of interfascicular cambium in the region of broad primary medullary ridge. The strips of interfascicular cambium join with the persistent fascicular cambium to form a complete ring of cambium. This cambium ring cuts secondary xylem towards inner side and secondary phloem towards outer side. It also produces secondary medullary ridge. In young stem, the medullary ridge are uniseriate, but later on they become multiseriate. The activity of cambium is differentiated or, or affected by seasonal variation, and therefore annual rings are formed. Each annual ring consists of thin-walled springwood and thick-walled autumnwood. The secondary xylem consists of tracheids, vessels, and xylem parenchyma. Student, that is the one of the gymnosperm plants in your syllabus which passages the vessels. And the other two plants that is cycas and pinus which are derived of the vessels. The xylem fibers are absent. The tracheids show the presence of single row of border pits in radial as well as tangential walls. They also show the presence of rims of sinew. The border pits are circular or elliptical in shape. The secondary xylem of Ephedra is characterized by the जो मैंने कहा अभी that is the presence of the vessels. Usually the vessels are not present in gymnosperms. The vessels are formed more abundantly in the springwood as compared to autumnwood. The vessels are larger and broader. Transitional stages showing the evolution of vessels from tracheids have also been noticed in the secondary xylem. A few young vessels show bordered pits in their end walls in which the middle lamella is lacking and the bordering area is narrow. Fully developed vessels showing perforated end walls also occur. The perforation pits in the end walls usually occur in groups. These are called foraminate type of perforation plates. The core cambium or the phalogen develops outside the secondary phloem during the third or fourth year of growth of the plant. It cuts core cells towards outer side and secondary cortex toward the inner side. So the pydrum consisting of core cambium, and secondary cortex replaces all the tissues outside the secondary phloem. Next, that is the leaf. The leaf is highly reduced and scale-like. Internally, it shows the many features or the following features. The outermost layer, that is the epidermis, which is consists of single layer of thick-walled cells and are interrupted at places by haplocylic type of stomata. Each stoma op- opens in an air chamber. The epidermis is followed by single layer of colorenchymatous palisade, followed by spongy parenchyma. Each leaf has two vascular bundles in the region of midrib. The vascular bundles are conjoined, collateral, and closed, with xylem facing towards adaxial side. Next, go to the life cycle. Reproduction. The Ephedra plants are heterosporous, that is, produce two kinds of spores, the microspores and the megaspores. The microspores are produced in microsporangia, born in male strobili, and the megaspores are produced in megasporangia, born on female strobili. Most of the species are dioecious. The male and female strobili are produced in male and female plants, respectively. But a few species are monoecious, that is, produced both types of the strobili, that is, male and female strobili. A strobilus resembles spike type of inflorescence and consists of an axis with decocately arranged strile scales and stamens or ovules. Unlike other gymnosperms, the strobili of Ephedra are compound, that is, the cone axis bears pairs of bracts which subtend either staminate or ovulate shoots. Male strobilus. The male strobilus is also called male cone, staminate strobilus, or male inflorescence. The male strobili are borne in whorls of 2 to 4 at each node of fertile branch of male plant. Each male cone arises in the axil of scale leaf. It bears about 2 to 8 or 12 pairs of rather thick bracts or scales arranged decocately on the short axis. The lower one or two pairs of bracts are sterile, whereas the upper ones are fertile. The male flowers arise singly in the axils of fertile bracts. Male flower or the staminate flower. Each male flower arises in the axil of fertile bract. It consists of a short axis which bears 2 to 8 microsporangia at its tip. The axis is called microsporangiophore or anthrophore. At the base of each microsporangiophore, two anteroposterior thin bracteoles are present which unite at the base to form a perianth. Each microsporangium is bi or trilocular and therefore called synangium. Next, that is the female strobilus. The female strobilus is also known as female cone or ovulate strobilus. It resembles with the male strobilus in its general appearance and place of origin. The female strobili are however smaller as a comparison to male strobili. They are borne in groups of 2 to 4 at the nodes of female branches, each in the axil of a scale leaf. Each female strobilus has a short central axis on which about 2 to 4 pairs of opposite decussate bracts are arranged. The bracts are fused below to form cup-like cupules. The bracts of female strobilus may be winged, dry, or fleshy and brightly colored. The lower pairs of bracts are sterile, whereas the upper two bracts are fertile. Each female bract bears a short-stalked female flower in its axil. Sometimes one female flower becomes abortive and the other assumes terminal position. Female flower. Each female flower is born in the axil of fertile bract. It consists of a short stalk and a terminal megasporangium. Students, before go to the megasporangium, we want to study about the mature microsporangium. The mature microsporangium is usually bilocular, sometimes trilocular. Each locule is surrounded by single layered epidermis. The middle layer and tapetum are used up in the formation of spores. Each locule is filled with a large number of microspores. The microspores are haploid, uninucleate, and elliptical in shape. At maturity, each locule opens by means of a terminal slit and releases the microspores. They are carried away by wind and therefore the pollination is anemophilous. Students will study about the microsporangium, megasporangium. The megasporangium or ovule consists of centrally located nucleus surrounded by two integuments. The bitagamic nature of ovule is unique among the gymnosperms. The outer integument, that is the perianth or the involucre, is formed by coalition of four segments giving it its shape of cup. It is morphologically equivalent to a pair of fused bracteoles. The inner integument is delicate and composed of two segments. The tip of inner integument elongates to form a micropylar tube, which is spirally twisted and exudes a pollination drop at the time of pollination. The lower half of inner integument gets fused with the nucleus, whereas the upper half remains free. The nucleus lies next to the integument in the central part of ovule. It forms a pollen chamber towards micropylar end. Students, tomorrow we'll study about the gametophytes. Now we end of today's topic. Thank you.